Hello everyone, this video is about T lymphocyte, T cell receptor gene rearrangement and generation of T cell receptor ligands. And this particular study note is developed by PG Pathshala and Childy. So I would like to express sincere gratitude to the development team. Then T lymphocytes, T cell receptor gene rearrangement and generation of T cell receptor ligands. The adaptive or acquired immune system combats pathogens with the help of very specialized antigen receptors. Adaptive immune system can provide long-term immunity against a particular pathogen and is very specific in its response. Therefore, it is not surprising that its antigen receptors are also highly specific in their interaction with the antigenic determinants of the pathogens. These antigen receptors are called either B cell receptors or T cell receptors depending on whether they are found on the surface of B cells or T cells. Like all the other receptors, the antigen receptors also have one domain that recognizes its ligand, or in this case an antigen, and other domain which transduces the signal into the interior of the cell. As would be expected, the antigen recognition domains differ greatly, while the signal transduction domains show very little variation between different antigen receptors. There are two types of TCRs, TCR alpha beta and TCR gamma delta. So TCR alpha beta receptors are comprised of two polypeptide genes, an alpha chain and beta chain. Similarly, TCR gamma delta receptors are also comprised of two polypeptide genes, an alpha chain and beta delta chain. So the T cells bearing TCR alpha beta receptors are much more numerous than those bearing the TCR gamma delta receptors. Further, the repertoire of antigen specificity of the TCR alpha beta receptor is also much higher compared to that of the TCR alpha delta receptor. It is noteworthy that the innate immune system also employs receptors to recognize pathogens. These receptors are known as pattern recognition receptors, or PRRs. The most widely studied PRR is a toll-like receptor, or TLR. Some of the major differences between receptors of adaptive and innate immune system are tabulated. Differences between pathogen receptors of innate and adaptive immune system, and BCRs and TCRs. So PRRs, there are PRRs in the sense pathogen receptors of innate and adaptive immune system. These are receptors of the innate immune system. They show limited variation in their pathogen specificities and are only few subtypes. They recognize pathogen specified molecular pattern, which are common molecules present on the surface of most pathogens. Since they recognize common molecules on the surface of pathogens, they are not pathogen specific. Their expression does not involve DNA rearrangements. Then BCR and TCR. These are receptors of the adaptive immune system. They show extensive variation in their antigen specificities. They recognize specific antigens on the surface of a particular pathogen. They are antigen specific and can even distinguish between closely related pathogens. Their expression involves DNA rearrangements between genes at the immune receptor loci, Ig and Tr loci. T cell receptors, or TCRs. As already discussed, these receptors are present on the surface of T cells. The initial experiments involving characterization and gene identification of TCRs were much more difficult than those for BCRs. This was due to a variety of factors, all of which delineate the differences between BCRs and TCRs. The main hurdles faced by immunologists while performing pioneering studies on TCRs were biochemical assays involved in the characterization of any macromolecule are easier to conduct if it is found in a soluble form. Since the TCR does not have a soluble variant, the biochemical assays were especially difficult to perform, a fact which was one of the main reasons for the delay in the characterization of TCR. Then antibodies and BCR can recognize the antigen directly. This simple antigen binding assays could be used to isolate and or purify the receptor for advanced characterization studies. The TCR, on the other hand, recognizes the antigen only when it is bound to the MHC molecule. Designing assays for such a system is extremely challenging and therefore makes the isolation and or purification of TCR more difficult. The plasma cells expressing antibody molecules transcribes immunoglobulin genes at high rate and produce many copies of mRNA encoding immunoglobulin genes. This facilitated the identification of Ig genes. On the other hand, the transcription of TCR genes in T cells occurs at a very low rate and produces fewer copies. Once again, antibodies and BCRs can recognize the antigen directly. Thus, simple antigen presenting binding assays could be used to isolate and or purify the receptor for advanced characterization studies. The TCR, on the other hand, recognizes the antigen only when it is bound to the MHC molecule. Designing assays for such a system is extremely challenging and therefore makes the isolation and or purification of TCR more difficult. The plasma cells expressing antibody molecules transcribe immunoglobulin genes at high rate and produce many copies of mRNA encoding immunoglobulin genes. This facilitated the identification of Ig genes. On the other hand, the transcription of TCR genes in T cells occurs at a very low rate and produces fewer copies of TCR mRNA. Thus, only a small fraction of the total mRNA of the T cell codes for TCR. This made the identification of TCR genes a very difficult endeavor. Then, now that we have a clear understanding of the inherent complexity involved in TCR related experiments, we can fully appreciate the clever and innovative ways in which early immunologists overcome these hurdles to investigate TCR in greater details. Characterization and isolation of the TCR. Some of the earliest studies regarding the characterization of TCR established the phenomenon of self-MHC restriction in TCR. In this regard, the studies of Zingernagel and Doherty played a crucial role. I'm not sure about the pronunciation of these scientists' name. Sorry for that. These researchers proved that the TCR of an animal recognizes the antigen only when it is bound to the animal's own self-MHC molecule. The earliest experiments regarding the isolation of the TCR protein were performed in the labs of Jim Allison, Ellis Reinherz, and John Kaplan and Philippa Marek. In their 1983 publication, Kaplan and Marek described the isolation of a TCR by generating monoclonal antibody against it. A brief and simplified description of their experiment is presented in its text. In this experiment, they immunized a mouse 1 with OVA, ovalbumin, and after a few days isolated T cells from this mouse. Then these isolated T cells maintained a subpopulation of T cells with OVA-specific TCR. These cells are short-lived and therefore to increase their lifespan, they were fused with the cells from a T cell tuber. The hybrid cells formed after such fusions are known as T cell hybridomas. So these T cell hybridomas were diluted and the clonal population from each different hybridoma cell was grown separately. Then the clonal population which was specific for OVA and hence not activated, studied for measuring IL-2 secretion is in response to OVA was injected into a new mouse 2. After a few days, B cells were isolated from the spleen of mouse 2. Some of the B cells generated antibody against the OVA specific TCR. These B cells were then fused to cells from B cell tumor to form B cell hybridoma. After these B cell hybridomas, the one that produced a monoclonal antibody against OVA specific TCR was identified. This monoclonal antibody was then used to precipitate out the OVA specific TCR from a solution containing all the membrane proteins found on T cells. This technique of using a specific antibody to precipitate out its antigen is known as immunoprecipitation. The importance of this experiment by Kaplan and Marek was that it allowed early immunologists to isolate the TCR specific for a particular antigen. This seminal experiment facilitated further investigation and characterization of the TCR. Mice immunized with OVA, T cells isolated from lymph nodes of mouse A, T cells from mouse 1, T cell hybridoma formation, mouse 2 immunized with the hybridoma having OVA specific TCR, hybridoma cells, B cells isolated from spleen of mouse 2, B cell hybridoma formation and selection of clone that produces antibody against OVA specific TCR. Then immunoprecipitation of OVA specific TCR using monoclonal antibody. So you will be getting monoclonal antibody against OVA specific TCR, TCR monoclonal antibody complex. Identification of the TCR genes. In 1984, two research articles were published back to back in the scientific journal Nature. Both these landmark discoveries revolutionized the field of immunology. Takmak's group discovered the gene sequence for the TCR beta chain in humans, while Davis and Hedrick's group discovered the same in mice. The strategy described by the latest research articles was especially elegant. The biggest hurdle in this study was the low expression of mRNA encoding the TCR. Davis and Hedrick hypothesized that mRNAs encoding the TCR must be found on membrane-bound polysomes. Identification of the TCR genes. In 1984, the Takmak's group discovered the gene sequence for the TCR beta chain in humans, while Davis and Hedrick's group discovered the same in mice. The strategy described by the later research article was especially elegant. The biggest hurdle in this study was the low expression of mRNA encoding the TCR. Davis and Hedrick hypothesized that the mRNAs encoding the TCR must be found on membrane-bound polysomes. Thus, by isolating only the membrane-bound polysomal mRNA from a T-cell hybridoma, they enriched their sample for mRNA encoding TCR. The researchers then prepared 32P labeled cDNA from this enriched mRNA. The enrichment was an important step as it reduced the number of cDNA clones to be screened later. B-cell hybridoma, isolation of membrane-bound polysomal mRNA, preparation of 32P labeled cDNA, B-cell hybridoma, isolation of membrane-bound polysomal mRNA, hybridization to eliminate non-T-cell specific cDNA, isolation of unhybridized cDNA, this was T-cell specific cDNA, southern hybridization to detect T-cell specific DNA rearrangement, identification and sequencing of cDNA clone encoding TCR. Next, they isolated membrane-bound polysomal mRNA from B-cell hybridoma. This was followed by hybridization of the radio-labeled cDNA from the T-cell hybridoma and mRNA from B-cell hybridoma. For the gene expressed in both T-cell and B-cell, the cDNA and mRNA hybridized together to form double-stranded structures. On the other hand, the cDNA molecule which remained single-stranded were those which are expressed only in T-cells. The single-stranded cDNA molecules were cloned. It was hypothesized that any cDNA, on the other hand, the cDNA molecule which remained single-stranded were those which are expressed only in T cells. These single-stranded cDNA molecule were cloned. It was hypothesized that any cDNA clone encoding a TCR gene would show a changed DNA profile in T cell compared to cells from other organs. Thus, certain hybridizations were performed with the DNA from various cell types using these cDNA genes as probes. This led to the identification of a DNA clone which showed different DNA bands on non-T cells and T cells. Further, when probed with cDNA from TM86 clone, T cells with different antigen specificities also showed band profiles which were very different from each other. These results pointed to the fact that TM86 cDNA is formed as a result of DNA rearrangement. Subsequent studies established that TM86 cDNA clone encounters for the TCR beta chain. Liver cells, T cells from different antigen specificities. Southern blot analysis of different cells using TM86 clone as probe. The genomic DNA of cells was digested by PVU2 enzyme. Liver cells from two different mice strains show the same band profile. On the other hand, the T cells with different antigen specificities show a great variation in their band profiles. Then organization of the TCR genes. The alpha and beta chains of the TCR beta. So, the alpha and beta chains of the TCR alpha beta receptor are encoded by TRA locus and TRB locus respectively. Similarly, the gamma and delta chains of the TCR delta gamma receptors are encoded by TRU locus and TRD locus. A common feature of all the chains of the TCR is that they contain a variable region and a constant region. The variable region of the TCR beta and TCR delta chains are encoded by 3 genes: V variable, D diversity, and J joining, while that of the light chains is composed of only 2 genes, V and J. On the other hand, the constant region of all the TCR chains are encoded by a single gene, the C constant gene. The TR loci in humans are located on chromosome 7 and 14. The TRA locus has the maximum span at around 100 kb, while the TRG has a minimum span at around 160 kb. The details of the human TR loci are mentioned in table. The number of these genes may vary among different individuals depending on their haplotype. For example, the TRA locus has 43 to 45, TRB locus has 40 to 48, TRG locus has 4 to 6, and TRD locus has 7 to 8 functional V genes. Therefore, further the number of TRBJ gene can be either 12 or 13 depending upon the haplotype of the individual. This indicates the relative positions of the functions. So, illustrate relative positions of the function V, D, J and C genes in the human TR loci as indicated on the IMGT database, International Immunogenetics Information System. Out of all the loci encoding the immune receptor gene, the organization of the TRA or TRD locus is the most unique. This is because at this locus, the genes comprising the TRA as well as TRD loci are found. Even more interesting is the fact that a few V genes at this locus can be used to encode both a TCR alpha and TCR delta gene. Such V genes are believed to belong to both TRA and TRD loci and are therefore denoted as TRAV, TRDV genes. After the V genes at the TRA or TRD loci, the V, J and C genes of the TRD locus are present. Although most of the V genes of this locus are present at the 5' end of the locus, the TRDV3 gene is an exception. This location is downstream or 3' of the TRDC gene. The J and C genes of the TRA locus are found downstream to the TRDV3 gene. The TRA VRD genes are located at the same locus with the VJ genes of TRD situated before TRA gene. Therefore, if the TRA locus gets rearranged, the VJ and D genes of TRD locus get excised from the germline DNA. Consequently, the rearrangement of TRA locus should preclude the formation of TCR gamma delta receptor. Concerned with this idea is the fact that the commitment of a T cell to TCR gamma delta lineage occurs much before its TRA loci are rearranged. The organization of the TRB is such that almost all the V genes except one, TRB V30, are clustered together at the 5' end of the locus. The V, J and C genes of the TRB locus form two separate clusters, VJCY cluster and DJC2 cluster. The location of TRB3V30 gene is peculiar, located at the 3' end of the locus. The TRG locus is quite similar in organization to the TRB locus. All the V genes at this locus are located at the 5' end. This is followed by JCI cluster. The JC2 cluster is located further downstream to the JCI cluster towards the 3' end of the locus. Structure of the TCR complex. All the TCR chains possess a variable and a constant domain. Another common feature of all the receptors is that they possess two key components. One of the components is responsible for ligand, in this case antigen recognition, while the other is responsible for signal transduction across the plasma membrane. The variable regions of all the TCR chains together form the antigen recognition site of the TCR. On the surface of T cells, the TCR is found in association with the CD3 complex, and the resulting complex is referred to as the TCR CD3 complex. The CD3 complex is involved in signal transduction and is composed of 3 polypeptide genes which are recognized as heterodimer, although they bear the same names. The gamma and delta chains of CD3 complex differ greatly from gamma and delta chains of TCR. The polypeptide chains of the CD3 complex contain long cytoplasmic tails that help in signal transduction. TCR CD3 complex cell membrane TCR. Comparison between TCR and BCR. The BCR and TCR differ greatly from each other in a variety of ways. TCR found on the surface of T cells, BCR found on the surface of B cells. Antigen recognition component is composed of either alpha and beta or gamma and delta chains. Antigen recognition component is composed of 2 heavy and 2 light chains, kappa or lambda. Then signal transduction component is CD3. Signal transduction component is Ig alpha or Ig beta. No soluble form has been reported for TCR. Soluble form of BCR called antibody is secreted by plasma cell. Contain 2 identical antigen recognition sites. Contain only 1 antigen recognition site. Encoded by genes of the TRA and TRB for TCR alpha beta or TRG and TRD for TCR alpha gamma delta loci. Encoded by genes of the IGH, IGL and IGK loci. Recognizes antigen only when it is bound to MHC. Recognizes antigen by itself, then does not undergo somatic hypermutation, so that once a TCR is formed in the thymus, it does not undergo any further change. Undergo somatic hypermutation so that the BCR can change its amino acid sequence after it comes in contact with its antigen. So TCR does not undergo somatic hypermutation, so that once a TCR is formed in the thymus, it does not undergo any further change. But BCR undergoes somatic hypermutation so that the BCR can change its amino acid sequence after it comes in contact with its antigen. Okay, then combinatorial diversity of TCR genes. The various V, D and J genes can combine in many different combinations and thus give rise to antigenic receptors with a wide variety of antigen specificities. This phenomenon is known as combinatorial diversity. The various V, D and J genes combine in many different combinations and thus give rise to antigenic receptors with a wide variety of antigen specificities. This phenomenon is known as combinatorial diversity and is known to occur at both TCR loci as well as Ig loci. The total number of maximum combinations possible for both TCR alpha beta and TCR gamma delta chains are calculated. So TCR gamma delta chains can sometimes consist of 2 D genes instead of 1 and this may have a slightly higher number of possible combinations than indicated, but it is still many orders of magnitude less than those for the TCR alpha beta gene. Then DJ combination, VDJ recombination and VJ recommendation. TRB and TRD loci contain V, D and J genes and undergo 2 separate combination events. One event links D gene to J gene and is called a V to J or VJ recommendation. The other event links the rearranged DJ genes to V genes and is called V to DJ or VDJ recombination. The sequence of recombination events at the TR loci is precise with V to J or VJ recommendation occurring before V to DJ, VDJ recommendation. Further, the TRB, TRG and TRD loci get rearranged before the TRA locus. The TRA and TRG loci do not contain D genes and therefore undergo V to J or VJ recombination only. As we will discuss in the later sections, the precise mechanism of molecular processes that lead to all these kinds of combination events are essentially the same. It should be noted here that the same process occurs during recombination of Ig loci also, a general term which describes all these types of DNA rearrangement events is somatic recombination. Thus, somatic recombination is the phenomena of DNA rearrangements occurring in the lymphocytes, which are somatic cells. This is in contrast to meiotic recombination, which occurs only in germ cells. Recombination signal sequence RSS. The precise sequence at which DNA is excited to enable combination events are highly conserved across most vertebrate species. Further, the sequences are similar between TR and Ig loci. These sequences are known as recombination signal sequence RSS. TRA, TRD locus, TRB locus, TRG locus. So as shown, RSS consists of a conserved 7BP heptamer sequence and 9BP nonamer sequence which flank a relatively non-conserved sequence. Depending upon the length of its spacer sequence, an RSS can be either a 12BP RSS or a 23BP RSS. As their name suggests, a 12BP RSS contains 12 BP long spacer DNA, while a 23 base pair RSS contains a 23 base pair long spacer DNA. An interesting feature of an RSS is that a recombination event can only occur between dissimilar RSS, a phenomenon known as 12 to 23 rule. Thus, 12 BP RSS can only recombine with 23 BP RSS and vice versa. The mechanism involved in TCR gene arrangement. The molecular processes involved in somatic recombination are the same irrespective of whether it is VJ, VDJ or VJ recombination. Moreover, the mechanism of somatic recombination at the TCR gene loci and at the IG loci is also essentially the same. Somatic recombination occurs in a highly ordered manner and can be divided into the following steps: RAG, binding and cleavage, hairpin loop and signal joint formation, P nucleotide addition, exonuclease activity, and N nucleotide addition. Then ligation of coding ends to form coding joints. RAG1 and RAG2 binding, cleavage by RAG1 and RAG2, 3' OH, end that launches nucleophilic attack on opposite strand. Ligation of signal ends by DNA ligase 4, coding ends containing hairpin loops, signal joint. RAG binding and cleavage. The first step in the process of somatic recombination is the binding of protein that cleave the target genes at their RAS. These proteins are aptly named the recombination activating gene protein, as their deletion mutants fail to undergo somatic recombination. So RAG proteins are specifically expressed in the lymphoid linkage and occur as a complex of two proteins, RAG-G1 and RAG-G2. RAG protein recognize the RSS of the target genes and introduce break in one of the two DNA strands at both target sites. This happens in accordance with the 12-23 rule. The breaks are produced at the junction between the gene and its RSS. Thus, a free 3' hydroxyl and a free 5' phosphate group are generated at one strand on each of the two sites. So, hairpin loop and signal joint formation. The free 3' hydroxyl group react with the 5' phosphate group in the backbone of their opposite strands, leading to the formation of two hairpin loops. This event generates a double-stranded break in the target DNA at both sites. Thus, two coding and two signal ends are formed. So, a signal ends joining together to form a signal joint, which is excised from the rest of the DNA and is lost during subsequent cell divisions. P nucleotide addition, exonuclease activity, and N nucleotide addition. Next, the enzyme Artemis cleaves the hairpin loop. Artemis. This cleavage can be asymmetric or symmetric. Asymmetric cleavage generates sites for P nucleotide addition, which is called so because the filling in reaction by DNA polymerase generates palindromic sequence. Simultaneously, two more processes occurs which along with P nucleotide addition increase the junctional diversity during recombination. One of these processes is the exonuclease mediated DNA cleavage. The other process is N-nucleate addition and is called so because it brings about non-templated addition of random nucleotides. The introduction of nucleotide variations by P nucleotide addition, exonuclease cleavage and N-nucleotide addition at the junction between two genes during somatic recombination is known as junctional diversity or junctional flexibility. Then ligation of coding ends to form coding joints. The next step involving filling in of the gaps between DNA polymerase. Finally, the coding ends are ligated to form a coding joint with the help of DNA ligase 4 and non-homologous N-joining NHEJ proteins. In the case of VJ and VDJ recombinations, this stage represents the end of somatic recombination. However, in case of DJ recombination, this stage is followed by initiation of recombination to link a combined DJ to a V gene PDJ recombination. Stage specificity of events during TCR gene rearrangement. The development of T cells occurs in thymus. The stage of T cell development has profound effects on the process of TCR gene rearrangement. The development of stages of T cells are studied with respect to expression of certain well-defined surface molecules. These surface molecules are usually designed CD, which stands for cluster of differentiation. CD4 and CD8 are among the most widely studied T cell surface molecules. Depending on the presence or absence of CD4 or 8 on their surface, the T cell development stages are defined as double negative or DN, CD4 CD8 double positive or DP, CD4 plus CD8 plus, and single positive or SP, either CD4 minus CD8 plus or CD4 plus CD8 plus minus. Of these three stages, the DN stage is the earliest stage during T cell development based on the presence or absence of two more cell surface molecules, CD25 and CD44. The DN stage can be further subdivided into DN1, DN2, DN3 and DN4 stages. Here you could see correlation between T cell development stages and TCR T rearrangement events. CLP, common lymphoid progenitor. The rearrangement of the TRB, TRG and TRD genes occurs at DN2/3 stages. Therefore, this is the time at which T cell fate decision occurs between TCR alpha beta and TCR gamma delta lineages. If the TRG and TRD loci get rearranged, then the cell gets committed to TCR gamma delta lineage. However, if a successful rearrangement occurs at the TRB locus, then the cell gets committed to TCR alpha beta lineage and combines with a pre-T alpha chain to express a pre-TCR on its surface. The expression of pre-TCR leads to allelic exclusion so that the other TRB allele does not rearrange a different TCR beta chain. The TCR beta lineage cell then reaches the DN4 stage and starts rearranging the TRA locus. The TCR alpha chain then combines with the TCR beta chain, therefore expressing a TCR on its surface. Then it reaches its DP stage. So later the T cell undergoes positive and negative selection and finally enters the peripheral circulation. Then regulatory factors involved in TCR gene rearrangement. Many factors affect the process of TCR gene rearrangement. Some of these factors have to be present on the same DNA to have an effect, are known as cis-acting factors. On the other hand, there are many factors which do not need to be present on the same DNA to an effect. Such factors are said to exert their effect in trans and others known as trans-acting factors. A few examples of cis and trans-acting factors are mentioned. Here, enhancers, promoters and RSS. Usually proteins such as RAG1 and RAG2, TDT, then DNA ligase 4, high mobility group 1,2 proteins and Ku proteins. Then generation of TCR ligands. The TCR recognizes its antigen only after it has undergone the necessary processing and is presented with the self-MHC molecule. Thus, it can be said that in case of T cells, both the receptor as well as its ligand have to be generated in a specific manner. This is the absolute contrast to the antigen recognition by BCR, which recognizes the antigen directly as it is. A single positive SP T cells can be either CD4 plus helper T or TH cell or CD8 plus cytotoxic T or TC cell. The TCR of a CD4 plus T cell recognize exogenous antigens. These antigens are processed and then presented in context of a class 2 MHC molecule. The class 2 MHC molecule are expressed in antigen presenting cells only. These APCs internalize the exogenous antigen via endocytosis or phagocytosis and process it by breaking it down into smaller peptides. These small peptides are then loaded onto the class 2 MHC molecules and subsequently are expressed on the surface of the APC. The TCR on the surface of Cl4 plus T cells interact with this peptide class 2 MHC complex and in combination with appropriate co-stimulatory signals activates the CD4 plus TH cells. A simplified flow chart depicting the major events is given below. Exogenous antigen internalized and processed by APC, peptide class 2 MHC complex, activation of TCR of CD4 plus TH cells. The TCR of CD8 plus T cell recognizes endogenous antigens which are processed and then presented in context of a class 1 MHC molecule. The class 1 MHC molecules are expressed in all the nucleated cells. The endogenous antigen is produced by altered self cells, which may be either virus infected cells or cancer cells. In the cytoplasm, the endogenous antigen is broken down into smaller peptides, which in turn loaded onto the MHC class 1 molecule present in the ER. This peptide class 1 MHC complex is then expressed on the surface of ASC. The TCR on the surface of CD8 plus T cells interact with this peptide class 1 MHC complex and in combination with appropriate co-stimulatory signals activate the CD8 plus T cells. A simplified flowchart depicting the major events. So, endogenous antigen produced by ASC digested into smaller peptides in cytoplasm, peptide class 1 MHC complex, activation of TCR of CD8 plus TC cells. Summary. The TCR alpha, beta, gamma and delta genes are encoded by TRA, TRB and TRG and TRD loci respectively. The variable A, B, D, G. Okay. The variable region of TRB, TCR, beta and delta chains are encoded by 3 genes, the variable gene, diversity gene and joining gene. The variable region of TCR alpha and gamma are encoded by 2 genes, variable gene and the joining gene. On the surface of T cells, the TCR is found in association with the CD3 complex and the resulting complex is referred to as a TCR CD3 complex. The CD3 complex is involved in signal transduction and is composed of 6 polypeptide genes. The T lymphocyte and B lymphocytes use the same molecular machinery to rearrange the TR and IG loci respectively. The TRB locus rearranges during DN2 and DN3 stage, while TRA locus rearranges during DP stage of T lymphocyte development. The RAG genes are essential for the process of somatic recombination, and their deficiency results in immunodeficiencies of both B and T cells. RSS are the DNA sequences where the RAG proteins bind the DNA to cleave it during somatic recombination. The TCR recognizes its antigen only after it has undergone the necessary processing and is presented with a self-MHC molecule.